So welcome back to module number 18. We are in unit 4 still of sensation and perception. So module 18 is about vision, specifically sensory and perceptual processing. Some really interesting stuff, but a lot of terms to remember. I'm talking a lot about rods and cones and things called feature detectors. In general, as an overview, we're talking about our eyes receiving light energy and transforming that light energy into neural messages. And then our brain creates what we consciously see. So think about that. It is just absolutely unbelievably amazing. Our eyes are seeing this light energy, and remember that through that process of transduction, we're turning it into neural messages. And then our brain is working to create what we consciously see. So the specific learning targets for this module are to discuss the characteristics of the energy that we see as visible light and describe the structures in the eye that help focus that energy. Also, to be able to describe how the rods and cones process information and explain the path information travels from the eye to the brain. Third, to discuss how we perceive color in the world around us. Fourth, to be able to describe the location and function of feature detectors. And 5, to be able to explain how the brain uses parallel processing to construct visual perceptions. So when you look at like a bright red tulip or something like that, the stimuli striking your eyes are not particles of the color red, but pulses of electromagnetic energy that your visual system is then perceiving as red. So what we're seeing is visible light is just a little slice of the wide spectrum of the electromagnetic energy. And that's what's shown in this figure right here. So the part of the spectrum visible to humans is actually quite small as compared to like birds. There's a really cool video on YouTube that has shows you what birds can see, like the different colors that they can see that we can't, and it's really fascinating. I'm a little jealous of those birds, but we can see, as humans, we can see light waves with a frequency of a little less than 400 nanometers and a little more than 700 nanometers. So what are the characteristics of light waves? What we see as light is, like I said, just a tiny slice of a wide spectrum. The wavelengths visible to the human eye, again, extend from the shorter waves of the blue-violet light to the longer waves of the red light. Waves vary in wavelength, which is the distance between the successive peaks. As you can see, the short wavelengths equals high frequency in bluish colors, long wavelengths equals low frequency, the reddish color one there. The shorter the wavelength, the higher the frequency. I lost my cursor there. Okay. Waves also vary in what's called amplitude, and that's just the height from the peak to the trough. So wave amplitude influences the perceived brightness of color. So wavelength is telling us what hue, which hue is the same as color, am I seeing? And amplitude is how bright is the color that I'm seeing. Okay, so that's the difference between wavelength and amplitude. So a little comprehension check on that very quick overview of wavelength and amplitude that we just went over. Say your best friend decides to paint her room in extremely bright electric blue. Which of the following best fits the physical properties of the color's light waves? So let's think back. So we're going to do extremely bright electric blue color. So if it's extremely bright and electric blue, let's back up a second. If it's extremely blue, bright and extremely blue, we're going to say that it's short wavelength and great amplitude. So short wavelength and large amplitude, that would be B. So let's look at the parts of the eye, the structures of the eye that help us focus our energy. And this will be something that you're really going to have to take some time to learn and understand. And I would even recommend drawing it out and practicing the different parts of the eye. It's important to know these basic parts of the eye for AP psychology. Basically, in general, light is entering, how light passes into your eye. Light is entering through the cornea, which bends light to help provide your focus. The light then passes through the pupil, which is the black part of your eye, which is that small adjustable opening. Surrounding the pupil and controlling its size is the iris, which is the colored muscle that dilates or constricts in response to the light energy. Sorry about my dogs barking. So what is the cornea? So the cornea protects the eye. If you wear contacts at all, they go on your cornea. The cornea is the eye's clear protective outer layer covering the pupil and iris. Light enters the eye first through the cornea. How about the pupil? The pupil is a small adjustable opening in the center of the eye through which light passes. Okay, sorry about that. I had to go try to get my dogs to stop barking. But so what is the pupil again? The pupil is that small adjustable opening in the center of the eye through which the light is passing. The light passes. How about the iris? Well, the iris is the ring of muscle tissue that forms the colored portion of the eye. So depending on what color eyes you have, I have green eyes, which is, I think, one of the rarest color eyes, if not the rarest color eyes to have. But it is the iris is the colored portion of the eye around the pupil, and it controls the size of the pupil opening by expanding and contracting over the pupil. So what's really fascinating about the iris is that each iris is so distinctive that an iris scanning machine can confirm your identity, which is really fascinating. You see that in movies often, that, you know, can they get into the secret room? They have to have their eyes scanned. So that is, your irises are so unique that is possible. So what is the lens? The lens is the transparent structure behind the pupil that changes shape to help focus images on the retina. Its purpose is to change shape and focus the image to the back of your eye. So how does the lens change shape? So to focus the rays, the lens changes its curvature and thickness in a process called accommodation. But if the lens focuses the image on a point in front of the retina, you see near objects clearly, but not distant objects. This is nearsightedness or myopia, and it can be remedied with glasses, contact lenses, or surgery. I have severe myopia, whereas, you know, being able to see well is considered 20/20. My vision is 20/1200, which is seeing at 20 feet what the average person sees at 1200 feet. Now, fascinatingly, with my contact lenses, I'm able to see 20/20 still, which is great. I've not had surgery, and according to my eye doctors, they always say that even if I did have surgery, I'd probably still need glasses because I am so nearsighted. So I have extreme nearsightedness. Hyperopia, on the other hand, which is farsightedness, is a result of the lens focusing light past the retina. So what is the retina? It's the light-sensitive, you can see it right here, the light-sensitive inner surface of the eye containing the receptor rods and cones, plus layers of neurons that begin the process of visual information. So again, these main parts of the eye are pretty important. So just take a minute here for, you know, less than a minute, 30 seconds, and just look at them and see if you could label them, and then the parts that are blank right there, and then I'll go over them with you. Okay, so up here at the top, where that one's pointing, would be the lens. Then this right here would be pointing to the pupil. You can see where that goes, hopefully. This one right here would be the iris, that colored portion. And this part out in front would be the cornea. And back here, that is the retina that's pointing to. So where's the retina located? It's located along the back of the eye and contains the sense receptors. sense receptor cells, the rods and cones that receive the incoming light waves. So this is a little bit about what happens within the retina. Light waves are transformed or transduced into neural impulses by the rods and cones and then pass to bipolar cells and ganglion cells. This is just a little image of what that looks like. Blown up really big, obviously. So the rods are retinal photoreceptors that detect black, white, and gray and are sensitive to movement. So you have about 120 million of them in your eyes. They're on the outer parts of the periphery of your retina, and they're really involved in night vision. They're necessary for peripheral and twilight vision when the cones don't respond. So some characteristics of the rods that you're going to want to know are that they're located on the outer part of the retina. They're sensitive to dim light and they enable black and white vision. They have no hotline to the brain. They share connections to a single bipolar cell sending a combined message to the brain. They're sensitive to faint light and peripheral motion. Now, on the other hand, the cones, think cones color, are retinal photoreceptors that are concentrated near the center of the retina, and they function in daylight or well-lit conditions. They are the receptors that detect the fine detail and create our color sensations. And we have about 6 million of those. And they cluster right around the fovea, which I don't think we've mentioned yet where that is, but I will show you. Some characteristics of cones. Again, they cluster around the fovea. In dim light, cones become unresponsive and we're unable to see color. Many cones have their own hotline. Singular cones have their own hotline to the brain. One cone transmits its message to a single bipolar cell, which relays that message then to the visual cortex, where a large area receives input from the fovea. So this is an interesting experiment to look at. How many dots can you see at one time? They're not really kind of dots. How many of those dark or black? If you focus in any of the 12 black dots and you see them, then you can see your peripheral vision won't allow you to see the ones around the outside. So I'm switching my focus on, it's kind of a freaky thing. Whatever you're staring at one of those black dots, then you don't see any of the rest of them because your peripheral vision is sort of shut off because you're focused. So the fovea, as I mentioned, we would talk about the fovea. It's the central focal point in the retina around which the eye's cones cluster. This is the area of greatest visual acuity or the sharpness of focus. You can see it back here. How about the optic nerve right over here? It's comprised of axons of the ganglion cells. It leaves through the back of the eye and carries those neural impulses from the eye to the brain. So the optic nerve is the sort of transportation system from the eye to the brain. What's that concept of blind spot? You've probably heard of the concept blind spot and wondered whether or not it's a real thing. Well, the optic disc is the point at which the optic nerve leaves the eye, creating a blind spot because no receptor cells, rods, or cones are located there. See where it is right there, where your optic nerve is going out? That is an actual blind spot that you have. So for a quick comprehension check, if you scratch your eye, which some of you may have done, which structure are you most likely to damage? If you think about it, it's the one that's going to be on the outside, the outermost part of your eye, which would be C, the cornea. So what happens to these neural impulses after they exit the eye? So the light energy is coming in and going through those different structures within your eye, comes through your optic nerve, and then what happens? So the optic nerve carries the impulse to the thalamus and on to the visual cortex. Remember, the visual cortex is in the occipital lobes in the back of your brain. So we've just gone over a ton of vocabulary, a ton of words, a ton of terms, a ton of anatomy. Make sure you understand those basic structures of the eye And like I said, what's really helpful for that kind of thing is to draw an eye and just label it. First, do it by looking at a picture of it, then do it by memory, because that'll really be able to test your retrieval and make sure that you're understanding and knowing exactly where those parts of the eye are. To learn how all the parts fit together, it may help to make rough sketches, like I said, and compare them. So, and this slide is going right along with the Myers Psychology for the AP course 3rd edition. So those would be the figures 18.3 and 18.4 if you have that textbook. That would be what it's referring to. And yeah, when you're making the rough sketches, don't worry about making it perfect. Just try to draw some rough sketches so you're kind of understanding, and you kind of go step by step to understand, oh wait, the light's coming in through the cornea and then the pupil and then the lens. Okay, just make sure you understand that process. So how do we see color? So we say that a tomato, for instance, is usually red, although there are tomatoes that are other colors, but is it really red? Is red in the tomato or is red in our mind? And so this is something really strange to think about. If no one sees the tomato, it's like if a tree falls in the woods, did it really fall if no one hears it? If no one sees the tomato, is it still red? So we say a tomato is red, but is it? The answer is no. First, the tomato is everything but red because it actually rejects or reflects the long wavelengths of red. The tomato's color is sort of our mental construction. The light rays are not colored themselves, as Sir Isaac Newton pointed out to us back in the 1700s. Like all aspects of vision, our perception of color resides not in the object itself, but in the theater of our brain. Okay, so it's all about what we're perceiving. So there are two different theories that we're going to go over. The first of which is the Young-Helmholtz trichromatic, which means 3-color theory. This is the theory that the retina contains 3 different types of color receptors. Remember, the color receptor starts with the C, cones. One most sensitive to red, one to green, one to blue. And when they're stimulated in combination, they can produce any color. So that's the Young-Helmholtz trichromatic. So remember, trichromatic, tri means 3, red, green, blue. Those are the 3 colors. So what about people who can't see color? Well, the photo on the left shows how people with red-green deficiency perceived a Buffalo Bills football game in 2015, and they were playing against the New York Giants. So we've got a green team and a red team. So everyone looks like they're on the same team if you're colorblind in this way. So if you were colorblind, red-green colorblind, you would see the teams playing like the picture on the left, which would be really frustrating because it'd be really hard to tell who's on which team. So the photo on the right shows how the game looked to those with normal color vision. So what are some characteristics of color blindness? It's pretty common. About 1 person in 50 is colorblind. Males are more affected because the defect is genetically sex-linked. Most people are not actually blind to all colors. They lack functioning red or green sensitive cones, or sometimes both red and green. So vision is monochromatic, 1 color, or dichromatic, and seems normal to them. Those are people that have color blindness. Again, things you need to understand about color blindness, because it is often a question that can be on the AP exam. It's important to remember that it's most prevalent in males and that red-green cone deficiency is the most common form. So the other theory that we're going to go over is the Herring opponent process theory. So Herring, after Young and Helmholtz came up with their theory, decided, well, this doesn't seem to cover everything. So Herring found that he thought photoreceptors were paired together to enable color vision. So red, green, blue, yellow, white, black. Activation of one color of the pair inhibits activation of the other. So for example, some cells are stimulated by green and inhibited by red. Others are stimulated by red and inhibited by green. And there's some fun little tests to kind of look at this theory. So we're going to try one to test out the opponent process theory. So what I want you to do is stare at this flag for a minute, and then we're going to move on to the next slide. Okay, we're going to move on. Now, what do you see? Yeah, that's crazy, isn't it? So that's some evidence for the opponent process theory. Did you see a red, white, and blue British Union Jack? Here's the answer for why the green became red. First, you stared at green bars, which tired your green response. Then you stared at a white area. White contains all the colors, including red. Because you tired your green response, only the red part of the green-red pairing fired normally. The same happened with the blue-yellow pair and the black-white pair. Hopefully that makes some sense, but it's a really neat little trick to do. It's fascinating. So how does color processing work? So first, we found out that the retina's red, green, and blue cones respond in varying degrees to different color stimuli, as the Young-Helmholtz trichromatic theory suggested. Second, we found that the cone's responses are then processed by opponent process cells, as Herring's opponent process theory proposed. So the Young-Helmholtz trichromatic theory shows that the retina contains color receptors for red, green, and blue. The opponent process theory shows that we have opponent process cells in the retina and the thalamus for red, green, blue, yellow, and white, black. These theories aren't in opposition to each other, they're sort of complementary, and they outline the two stages of color vision. The retina's receptors for red, green, and blue respond to different color stimuli, and the receptors' signals are then processed by the opponent process cells on their way to the visual cortex in the occipital lobe of your brain. So what are feature detectors and where are they located? Well, feature detectors are nerve cells located in the visual cortex of the occipital lobe that respond to a scene's edges, lines, and angles. They're highly specialized cells that process information in the brain. So feature detectors receive information from individual ganglion cells in the retina and pass it to other cortical areas where these supercell clusters respond to more complex patterns. So this is USA soccer player Abby Wambach, who in 2011 had one of many amazing goals. I think she's the highest goal scorer, male or female, for the US ever. And interestingly, I just recently met her a few days ago. But she was amazing. Her skills to be able to, in the air, to be able to kind of see and detect everything going on and be able to accurately redirect the ball and score so many goals via headers was unbelievable. And she was able to instantly process visual information about the positions and movement of Brazil's defenders and goalkeeper and managed to get the ball around them all and into the net. So she had some of these supercells going on for sure. And that is really hard, right? I played soccer in high school and in college, and her skills in this area were amazing. So Abby's a good example of that. So what is parallel processing? Parallel processing is thinking about many aspects of a problem simultaneously. This is the brain's sort of natural mode of information processing for many functions, including vision. So here's a little visual of how parallel processing operates. The brain delegates the work of processing motion, form, depth, and color to different areas. So if you look at this picture here, you've got the first one is motion you're looking at, form, depth, coloring, and these are all happening at the same time. After taking a scene apart, the brain then integrates them together, these subdimensions, into this perceived image that you actually see. How do we recognize faces using this parallel processing? So to recognize a face, your brain integrates information projected from your retinas to several visual cortex areas and compares it with stored information. And then you're able to recognize faces using your fusiform face area, a specialized area in your brain, and you can recognize like your grandmother. Sometimes supercells, actually nicknamed grandmother cells, do appear to respond very selectively. Whoops, Siri thought I was talking to her. That's strange. I don't know what triggered Siri, but we were talking about supercells and grandmother cells. Really interesting stuff about how we have specialized areas within our brain, specialized area within our brain, fusiform face area to recognize faces. Okay, so what would you answer? Which perceptual process explains why you can see varied aspects of your favorite singer's face and instantly recognize him or her? So we just mentioned that. That would be E, parallel processing. So overall, in the end, how do we actually see? Well, as you read, I'm just going to read this. It's kind of complicated. As you read these words, the letters reflect light rays onto your retina, which triggers a process that sends formless nerve impulses to several areas of your brain, which integrates the information and decodes its meaning. The amazing result is we've transferred information across time and space. And so from one mind to another, basically, and this all happens very instantaneously, effortlessly, and continuously. And it is indeed awesome. It's absolutely amazing when you're thinking about this light coming into our eyes, going through all those different structures, going through our optic nerve, becoming neural impulses, connecting with our visual cortex, being able to be recognized as special things by supercells, grandmother cells, recognizing faces through our fusiform gyrus. It's all pretty unbelievable, amazing stuff that enables us to see. So let's review our learning targets. The first one, discuss the characteristics of the energy that we see as visible light and describe the structures in the eye that help focus that energy. So the hue we perceive in light, which is the color, depends on its wavelength, and its brightness depends on its intensity. After entering the eye through the cornea, passing through the pupil and iris, and being focused by the accommodation of the lens, light energy particles strike the eye's inner surface, the retina. So describe how the rods and cones process information and explain the path information travels from the eye to the brain. So rods and cones convert that light energy into those neural impulses that are then passed on to our brain. Cones are found in and around the fovea, rods on the outer, the peripheral regions of the retina. Cones connect to one bipolar cell, rods share a bipolar cell. Cones are sensitive, remember cones color are sensitive to detail and color, rods to faint light and peripheral motion, things on the outer part of your visual periphery. Impulses move through bipolar and ganglion cells through your optic nerve, remember that's coming out of your eye, and can create a blind spot there, okay, where your optic nerve is, then to the thalamus and onto your visual cortex and your occipital lobe. So how do we perceive color in the world around us? We went over two different theories. The first one, the Young-Helmholtz trichromatic three-color theory, which states that the retina contains three types of color receptors, each most sensitive to the wavelengths of one of either red, green, or blue. And then there was Herring's opponent process theory to sort of explain some of the stuff that Young-Helmholtz did not. The neurons in the retina and the thalamus code the color-related information from the cones into pairs of opponent colors, red versus green, yellow versus blue, and black versus white. And the color processing occurs in two stages. So describe the location and function of feature detectors. Well, feature detectors are those specialized neurons in the occipital lobe, visual cortex. They respond to specific aspects of the visual stimulus. They receive information from individual ganglion cells in the retina and pass it to the other cortical areas where these supercell clusters respond to complex patterns in the environment. Think of Abby Wambach, specifically knowing how to respond as the ball is coming to her, going to head it into the goal past the goalkeeper in the World Cup. So explain how the brain uses parallel processing to construct visual perceptions. Through parallel processing, the brain handles many aspects of vision, color, movement, form, and depth, all at the same time, simultaneously. Other neural teams integrate the results, compare them with stored information, and that enables our perception, right? Think about all these things are going on in your brain right now. As you're looking at things, as you're taking information in, this is what is happening. Really fascinating stuff. Thanks so much for listening and watching. Please consider subscribing to my channel so you'll know when I upload new presentations or new information for AP Psychology or other psychology classes. Thanks so much. Take care.